Hello everyone. So, welcome to this program of Approach to Anthropology, and in this program we are going to talk about human genetics. As usual, we are going to take one question, and today's question I will take epigenetics and gene expression. Many of you might not have heard about this topic, but it is very important that how the non-coding factors on your DNA are important in gene expression, or you can say even other factors you can say, which is environmental factor, behavioral factor, how they influence the gene expression, right. Normally what you know that genetics or the gene expression is the product of a particular sequence of DNA, right. So, first I will give you an overview about genetics, what you all study, right, and especially our purpose here to understand human genetics, because we study anthropology, right, the systematic study of mankind. Now, see genetics is very important, and many of you people fear that genetics will be tough, right. It is not tough, it is just that you did not apply your right approach when you study genetics. Now, in genetics you study primarily heredity and inheritance, right, at the individual level, and this inheritance of the traits as well as diseases you cover. Right. So, in a sense you talk about the genetic diseases, right. Some of these diseases you know that may be because of some particular gene alterations or chromosomal alterations, right, that you study here. You also study the population genetics, right, where we talk about the distribution of genes, or you can say allele, in a population level, right. You study here polymorphisms, right. These polymorphisms can be useful to identify any individual or any population. Okay. You study in this Hardy-Weinberg law in order to understand whether a population is evolving or not, right. Although our syllabus of human genetics is limited, but there are many other branches like developmental genetics, molecular genetics, right, and everything we try to cover in such a lucid manner that you can understand each and every concept, right. Now, since this topic is very vast, this human genetics is very vast, we are going to cover one single topic, that is epigenetics, right, and how it is related to gene expression in today's class. Okay. So, our topic for today is epigenetics. Epi means above, or you can say beyond genetics, right. For understanding this, you must understand that how our phenotype that you see, this body type, is it is a product of the genes, right. The genes are the sequence of nucleotide, you can say. These are particular sequence of DNA, right, and this gene code for a particular trait that we all know. That means this trait, this is the visible trait, right. It can be anything, the skin color, it can be eye color, it can be hair, it can be anything that in our body, right? Some things are product of multiple genes, some thing are product of a single gene, right? So normally we know that gene is responsible for any particular trait, but here what we are going to understand that there are certain other things, right? Your gene demands something else, but that expression is sometimes either hidden or activated. That means these genes can be switched on or they can be switched off by some other mechanisms other than what is coded, right? Normally in simple language, if I say that your software determine your hardware, that is what you normally say, right? But there are certain other factors that can influence that software. That software will not represent the same hardware every time. That is what you can say in simple way, right? So gene may not be responsible for the same trait every time. There can be other external factors, right? Other than the coding, right? So these external factors can be environmental, or behavioral factor, right? When I say software in your simple language or the hardware, software means your genetic code, right? Genetic code is responsible for this body type, this phenotype. That is the normal thing. But what I say that there are some other factors that can influence the expression of this gene, right? And those factors we study here in epigenetics, right? So what it says, epigenetics is the study of how your behavior and environment can cause changes that can affect the working of your genes, right? Okay, so this is epigenetics. Then there is another term that I wanted to tell you here itself, that is epigenomics. Epigenomics. Now, what is epigenomics? See, in the way here we are talking about the epigenetic changes, right? That means, see, the gene was responsible for a particular trait, right? This single gene was responsible for any particular trait. That was the usual thing. But there were some environmental and behavioral factors which lead to different trait, right? Different. This gene will not represent the same trait now being under the influence of these factors, even though code is same. Okay, that is epigenetics. And all these epigenetic changes in the individual or in the genome, they are under epigenomics. So the totality of these all, or you can say all these epigenetic changes in any one individual is studied under epigenomics, right? So the difference is same, gene and genome, right? Gene is a single sequence, right? And genome is the totality of all genes that are present in a cell, or you can say in your entire body, right? So gene and genome, so similarly epigenetics and epigenomics. Now, what is the region that we are studying? See, anything that happen in your body is the product of gene expression. This is the expression of the gene, right? Gene is expressed in form of any trait, right? So epigenetics influences gene expression, and that may sometime result in some diseases, sometime it affect the growth, sometime it can you can lower down your immunity, sometime it can be responsible for cancers, right? That is why we are studying. Now, coming back to this epigenetics. So I can say that epigenetics is responsible for gene expression, right? Or you can say it can influence the gene expression. Right? Okay. Now, basically, how does it work? First thing first, that you know the definition of this now, right? That those changes which you can attribute to behavioral or environmental factors in the expression of gene, that is epigenetics, right? Then how does it work? What is the reason behind this, right? Or what are the causes of the epigenetic changes, right? So scholars say there can be 3 types of changes, right? Mainly there can be more, right? But we will talk about 3 major things. First is DNA methylation. The second is histone wrapping, right, or histone modifications. And third is non-coding RNA. See, to understand that how does epigenetics work, you must understand all these 3 things first, right? That DNA, it has methyl group, right? And this level of methylation determine the gene expression. The higher the level of methylation, the gene expression will be switched off, right? You can say higher methylation, higher levels of methylation, it is responsible for switching off any gene. ये स्विच ऑफ कर देगा, राइट? वो मिथाइल ग्रुप जो है वो जीन के एक्सप्रेशन को रोक देगा. देन लोअर लेवल ऑफ मिथाइलेशन, it can switch on, right? So methylation influences. Now, for example, methylation you study methylation somewhere else also, but I just give you. See, the expression of Huntington chorea disease, right? This is an autosomal recessive disease. This Huntington chorea disease, see, if this gene is inherited from the father side, then the expression is in the early age, and if it is inherited from the mother side, then the expression is at the delayed age, right? Because the mother's germ cell DNA is highly methylated, okay, as compared to the father side. Male DNA is less methylated than the female germ cell DNA. That is what you have to understand, and that is why you can say that DNA methylation, it can influence the way genes express themselves. Okay. Second is histone modifications. See, histone is a protein that is part of our chromosomes, right? And you can simply understand that chromosomes are made up of 2 things: DNA, deoxyribonucleic acid, and histone proteins. See, if you take, this is a DNA, this is a chromosome on the metaphase stage. So in this chromosome you will find that DNA is packed with the histone proteins, right? DNA is packed with the histone proteins. They are folded in such a close packed manner that both are here. Sometimes the histone, it packs DNA so tightly right, that it overfolds sometime DNA, right, and that folding, these modifications in histone, that also is responsible for switching off any gene, right. ज़्यादा folding हो गया, that means it will switch off. कम folding हुआ, it can switch on, right. You can say जरूरत से ज़्यादा folding, it can switch off. जरूरत से कम folding, it can switch on. So that can also be responsible for the epigenetic changes. Now, third thing that you have to understand is non-coding RNA, right. See, you have to understand what is the function of RNA first before understanding what is non-coding RNA. See, we have RNA which is responsible for transcription and translation. See, you have to first understand before going into this what is central dogma, right, and this is a mechanism through which you can say your genes express normally, right. These are three things. We cannot talk about everything in single 15-20 minutes class, right. So I am just telling you the glimpse of it. Central dogma is DNA, right. It converts, or you can say it make a mRNA, messenger RNA, that through a process which is called as transcription, right, and this messenger RNA is a coding RNA, right. It is a coding RNA because it codes for a protein, right. This process takes place inside the nucleus of the cell, right. Here we have DNA and here this mRNA is formed, right, and this mRNA carries a message. This mRNA will go out in the cytoplasm, right, and attach with the ribosomes to make a protein molecule, right. This is central dogma, and you can say this is how gene expression take place, because it is the proteins which are responsible for the trait, right. Gene expression take place through these proteins. Okay, so non-coding RNA क्या होता है, right. See, this was the coding RNA. There are certain RNAs which are non-coding, right. What happen? These non-coding RNA, they attach to the protein making sites, right, or they can just attach to the sites and they can switch off the gene, right. Their expression is stopped. That is how these non-coding RNAs work. So this is also responsible for this, right. Now, what are the factors, the environmental or behavioral factors, you can say, that can influence, right. See, first thing first is simply epigenetic changes are associated with our development, right. Okay, so factors. See, how development, first thing you have to understand is that when we are in the zygotic stage, right, every human is a product of one sperm and one ovum, right, and this zygote, it is totipotent, right, it has the potential to differentiate into any kind of cell. This zygote can, which is product of one ovum and this one sperm, right, this zygote have the potential to differentiate into any type of cell. Now, in our body, you can say this zygote is resulting into a baby, and this baby have nerve cells, this baby have muscle cells, this baby have bone cells, right, you can say heart cells, skin cells. Now, this baby have these different types of cell, but every cell, now first thing you must know, I believe that all of you know that cell is the structural and functional unit of our body. That means every cell, every tiniest cell contain all the genes that is required for your body, right? All genes are present in all cells. That is why we call genome, right? If I have to study your genome, I can take any cell, whether it is skin cell, whether it is nerve cell, whether it is your hair cell, or any cell that will represent your genome. That means all genes are there. All genes that are found in your body are found in any single cell that belong to any part of your body, right? That may perform any kind of function. That you must understand. Now, since this zygote is there, right? जाइगोट से सेल्स डिफरेंशिएट होकर डिफरेंट तरह के बॉडी पार्ट बने, राइट? बट नाउ सेल परफॉर्म डिफरेंट फंक्शन. दैट मीन्स जीन्स आर एक्सप्रेसिंग इन अ डिफरेंट वे. हियर इन नाउ सेल्स some genes which are responsible for this muscle contraction, they are switched off, you can say, right? Now cells, only those genes are expressive which are needed for nerve, this signal transmission, right? In muscle cells, there are certain genes which are switched off. In the bone cell, there are certain genes which are switched off and certain which are required are switched on. So don't you think this is epigenetics? Here, you can say human development. In this process of human development, right? Some genes are switched on and some genes are switched off. So it is not just a factor, rather it can be useful to understand this development also, right? Second factor, I believe you have got the idea about it. Second is age. See, with increasing age, the level of DNA methylation decreases, right? How we will understand this, that how this DNA methylation or the level of DNA methylation reduces? It is based on a study. Now, normally I just told you a few minutes before that women have higher levels of methylation. Women's germ cell, you can say especially, right? They have higher level of methylation. Germ cell DNA, you can say. DNA methylation. Why? See, for that you must understand one more major fact. That is that all the ova, right? Ovum जो होता है women का, all ovum of a woman, they are produced when that baby girl is still in the womb, right? See, all those were produced when she is still in the womb, मतलब उसका birth भी नहीं हुआ तभी उसके सारे ovum बन गए, right? These ovum are frozen in metaphase 2 of meiosis. In that stage, it is frozen. Within the mother's womb, it is frozen there. That means she is biologically a girl with carrying all the ovum that she will release when she will grow old, right? And starting with the menstruation up till the menopause, right? सब कुछ बन चुका है उसी टाइम पे. Now, the girl is in the womb of the mother, right? Now she is affected by various kinds of factors within the mother's womb, mother's hormones, mother's chemicals, or the overall environment of the mother's womb, right? Second, she will start releasing these ovum at maybe at 12 or 13 years of age. So she has to protect those ovum for 13 years. So don't you think that she require higher level of protection? So this higher level of methylation in women is a genetic adaptation which is selected by the nature, right? So nature ने ये select कर लिया, क्यों? क्योंकि because women require to protect these ova for at least 12 to 13 years, and at the most she required to protect her ova from the mother's hormones when she is still in the womb. So this is a selective advantage to the women. That is a known fact that you must know. This is given in all books, right? Now, this level of methylation, this reduces with age, even in the women. That is a study, right? And this study shows that 100-year-old women have lower levels of methylation, and a 26-year-old women have higher level of methylation. So you can say the methylation reduces with age. So this is a very important factor that is responsible for the epigenetic changes. So age is a very important factor that is responsible for epigenetic changes. Okay. Then third factor is your behavior, whether you are a smoker or non-smoker, right? So smoking can also result in epigenetic changes, right? Some genes, right, certain parts of a gene have lower level of methylation among the smokers, right? Once a gene is AHRR gene, this gene, right, certain part of this gene have less methylation in smokers than the non-smokers. So you can say smoking can alter the expression of the gene. So this is another factor. Then there can be certain infections that can alter the expression of a gene. For example, fourth factor is disease. Which disease? TB, tuberculosis, right? It can also alter the expression of any particular gene. You can understand that those with tuberculosis, they have lower immunity because of some changes into the histone protein, right? TB causes changes, tuberculosis, it leads to modification in the histone, right? And that is responsible for turning off one gene which is known as IL IL-12B gene, right? And this is responsible for lower immunity among the TB patients. That means they are susceptible to infection. This is another factor that can influence the epigenetic changes, right? Then there are other factors, right? Nutrition also can sometimes result into the epigenetic changes, right? Nutrition can be another factor that can be responsible for the epigenetic changes, right? It was observed that people who suffered malnutrition, okay, or you can say the children born to the mothers who suffered malnutrition during the you can say the period of fetal development, then these people, the children, they can suffer from schizophrenia, diabetes, heart disease, etc. So these are some factors that can be responsible for the epigenetic changes, right? Okay. Now what are the effects or consequences, you can say, right? See, consequences are visible here only, right? First thing is some epigenetic changes can be positive that are required. One thing is that human development, right, and all these things. You can say epigenetic changes can also be responsible for aging, right? So the consequences, you can say, of these epigenetic changes. Okay, see, some changes are responsible for normal growth and development. For example, development में हमने जो देखा, right? So the consequences are not always bad that you must keep in your mind, right? All epigenetic changes are not bad, because what we see, there are many factors in our body which are not coded by any particular sequence of DNA, right? So consequences are not always wrong. Your mind will always read like cancer, your lower immunity or any such thing. No, there are developmental factors. You can say it is also associated with aging, right? That also we have seen. So we can better understand, you can say this aging can also be attributed, because there are many theories regarding aging, right? Some say it is due to the genetic blueprint, but only genetic blueprint cannot completely tell us about the process of aging, right? So aging can be a result of this epigenetic changes also along with other factors. That is one thing, right? Second thing, it can be responsible for the normal growth and development. That we have seen when we were talking about the factors, right? Then negative consequences, you can say certain diseases, or you can say especially the genetic diseases caused by sudden alterations, because if some gene are they turn off or turn on, that may cause a cancer, right? You can simply say that this BRCA1 gene, right, it can be responsible for the breast cancer, right? BRCA gene. So it can be responsible for some cancers, some form of cancers, genetic diseases, right? Sudden mutations can occur, right? So even sometime this epigenetic changes can contribute to, you can say, the human evolution also, or you can say simply evolution, right? Now finally, after writing these 4 points, definition, causes, factors, consequences, you have to conclude using the significance of this understanding of epigenetics. See, earlier the geneticists, they think that every expression in the body is a result of some coding, but now geneticists know that out of 100% of our DNA, only 2% is just coding, right? That means only 2% of your DNA code for some gene, or you can say it is responsible for the phenotypic expression, or you can say gene expression, or you can say responsible for trait, right? And this 98% is non-coding, right? So now we know that there are many factors that can affect this 2%, and that is where the significance of epigenetics comes into play. So that is all I have to tell you. Again, let me tell you that what can be the structure: definition, causes, factors, consequences, and conclusion. These 5 things, if you write in this, right, you can address any question. If suppose there is a question, epigenetics and gene expression, that also can be very easily addressed if you have understood the entire mechanism that I have discussed here. Right. So I believe many of the concepts you might have got from this single session, right? If not, you can write back to us, and we will definitely try to help you. And keep on watching. We will meet again. Happy Diwali and stay safe, right? All the very best.